Good afternoon, everyone. My name is SNagrath. I'm an associate professor in Department of Chemical Engineering, and I'm also the PI, one of the PI on the spatial analysis program. Today, I'm going to talk about rare cell isolation and single cell analysis. Most of the time when we talk about single cell analysis, we are trying to identify the heterogeneity in bulk cell populations that are more abundant. But how about if you have a complex fluid and you want to understand the heterogeneity of a particular rare subpopulation? For that, we try to develop a workflow starting from a complex fluid, enriching the target population, and then performing the single cell analysis on those rare target cells. There are a lot of studies up and coming in these days along these lines, and we are presenting a workflow that we have developed at the University of Michigan. For the rare cell isolation, we are using here a size-based microfluidic labyrinth device. It isolates the cells based on their size. So as you can see, if the sample is inputted into this structure, the sample goes through this complex curvature and the cells get sorted by their size. You can see in this high-speed video, the larger cancer cells, the tumor cells, which are larger in size compared to the WBCs, they get streamlined into a single streamline and they are being collected into the second outlet in the device, whereas the smaller WBCs, again, you can see in the top, they are streamlined into a single file and they are collected being through the first outlet. So just purely based on the size, we are able to isolate the cells with a greater than 90% efficiency. And here is a demonstration of CTCs that are isolated from a different breast cancer patients. We are not only able to isolate the cells, but these cells are live. So you can sort these cells into single cell suspension using a fluid MC1 machine or some other workflow, and then you could analyze them at a single cell level. So today I'm going to present two different workflows. Here is an example of isolation of lung cancer cells first, and then we wanted to do a multiplex analysis at a single cell level. So here is the CTCs isolated from 25 lung cancer patients. These are all the patients having to have some targeted mutations in EGFR gene. So we have analyzed these CTCs both phenotypically and genotypically. Here you can see the CTCs that are present in the blood that have EpCAM positivity or Vimentin positive or pan-CK positive or triple positive cells. So we're not only able to isolate the cells in significant numbers, but we can also follow the CTCs through the treatment cycles. So you can monitor the phenotypic variation in these subpopulations as the patient undergoes treatment. We are not only able to isolate the single cells, we can also isolate clusters of CTCs, and we have shown that these clusters of CTCs presence have a prognostic significance. Besides for this topic, but still we can expand the CTCs also that are isolated from the microfluidic labyrinth and then culture them and expand them and use them to screen for any targeted therapies. So we have taken those cells that are isolated from lung cancer patients using the microfluidic labyrinth, and then we sorted them into single cells using the fluid MC1 machine. So once the cells are sorted into single cell suspensions, we prep the cDNA, and then we split the cDNA into two different workflows. So one of those workflows is we wanted to identify the targeted mutations at the single cell level in these CTCs. So we took the cDNA and then we subject this to PCR, targeted PCR in droplet digital PCR, ddPCR, rain dance machine in this particular example. So we could analyze at a single molecule level if there are any targeted mutations that are present. And the other workflow is taking a part of the cDNA, and then we did a multiplex PCR, qPCR, to look for 96 genes expression on 96 single cells at the same time. So using this workflow, this is an example of the data that could be generated. Here are the CTCs that are isolated, and these single cells were subjected to targeted PCR using the ddPCR, and you can see that the patients express a different type of mutations, and we were able to detect the same mutations using the ddPCR analysis. Here is the expression profile of the same CTCs. Here is for different CTCs from a same patients, and how their expression varies across these patients, across these different cells. For example, vimentin is heterogeneous across different cells from the same patient. At the same time, using the ddPCR, we are also able to establish both L858R, CT790M, the resistant mutation, as well as a deletion in exon 19. And the different single cells from the same patient have different levels of this mutation that is expressed. So we could really count the number of copies that are mutant within these single cells, along with the gene expression analysis. The other type of analysis that we would like to do was sequencing. Can we take the cells that are isolated from microfluidic labyrinth, and then can we subject them to sequencing at a single cell level? So we developed a workflow using the depth array. Depth array sorts the cells based on the fluorescence. So you can stain the cells. So the cells are purified in labyrinth, and then they're fluorescently labeled, and then they're loaded into the array cartridge. This cartridge really works on the basis of dielectrophoresis. So they create the cages of these dielectrophoresis where the single cells are trapped, and then you can select the cells of your interest and then sort them into single cell suspensions, or whatever the groups of cells of suspensions you want. So how the depth array works is that you first load the cells into the main chamber, and there are around 80,000 depth cages actually within this chamber. So the cells are trapped within these depth cages, and then based on their fluorescence, you can select the cells of your interest among 80,000 cells and move that one cell at a time into this parking chamber. And from this parking chamber, you can then release one cell at a time into your tubes or a well plate. Here is an example of how we are moving a cluster of cells across the depth array. So once the cells are sorted into different cages, you can move the cells selectively into a parking chamber, just like as shown. This can be done manually and automatically too, based on the parameters that you specify. For example, based on the fluorescence, diameter, area, you can give different parameters to be able to sort. So here is the workflow for the CNV copy number variation analysis of circulating tumor cells that we did using this labyrinth depth array workflow. So after processing from the labyrinth, the cells are loaded into depth array, and then the single cells are sorted, and then we did a whole genome amplification, and then do a library prep, and then we sequenced with a low pass genome sequencing. So here are the cells, how they appear on the depth array. These are some of the cells we sorted. We sorted some circulating tumor cells along with the WBCs as a control from the same patients based on their fluorescence. So we enriched the population from labyrinth, and then we stained to select the specific subpopulations using the depth array. And then we collected those single cells, and then we did a low pass genome sequencing. Here is an example of single cells from a pancreatic cancer cell line, as expected, which matched to the already published data that we were able to establish the copy number gains and the losses within these different cell lines. And when we looked at this is an example from a pancreas CTCs and a WBC isolated from the same pancreatic cancer patients. And you can see that CTC specifically or positive for copy number variations. This particular CTC has a gain in a different number of genes, and also loss in a different gene, which were both were absent in WBCs. And I hope with that, I was able to show you that we are not only able to pre-enrich the rare cells prior to single cell analysis, which could save the money and the resources, as one will be sequencing what exactly they need rather than all the cells that are present. And also rare cells can be isolated based on the size, which is what I demonstrated here. But we can also isolate the cells based on the immunoaffinity based strategies like targeting a specific protein on the surface. Or we can tailor make these isolation based on the application. I hope I have shown that the feasibility that we could work with the live cells or fixed cells. The example that I showed was with the fixed cells, but we are able to do both the live cells and fixed cells using this labyrinth and Depore workflow. And we have established the workflows to analyze the cells, these rare cell populations. I have shown an example of circulating tumor cells, but we can do this for any specific subtype of T cells or NK cells or macrophages. And then we can do downstream analysis that includes either a targeted PCR using the digital droplet PCR, which has a single molecule resolution, or you can do a multiplex PCR for multiple genes at the same time, or a targeted sequencing like I have shown with the Depore. If you have any questions or comments, I will be happy to answer them. Please feel free to reach us. With that, thank you very much for your attention.